Hello everyone, and thank you for attending today's webinar, Amphetamine to Zolpidem, Navigating the ABCs of Early Phase CNS Drug and Cannabinoid Development. I'm Sharon Saltz, editor of NeuroCentral at Future Science Group, and I'll be your host for today's event. Before I introduce you to our speaker, I'd like to quickly cover a few housekeeping items. Firstly, all the widgets are resizable and movable, so please feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. Please let us know your thoughts on today's webinar by tweeting us at neuro_central, the hashtag NCWebinars, or simply share today's webinar with your friends and colleagues using the Share This widget on your screen. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. An on-demand version of today's webinar will be available approximately 1 hour after the webinar and can be accessed using the same audience link that was sent to you earlier. So, we are delighted to have Dr. Beatrice Setnik presenting for us today. Dr. Setnik joined Altasciences in 2019 as Chief Scientific Officer. In her previous roles as Vice President of Scientific and Medical Affairs at INC Research and CNS Health, Senior Director, Clinical Sciences at King Pharmaceuticals and Pfizer, and Research Scientist, formerly known as Ventana/Decision Line Clinical Research, she has accumulated impressive experience in early phase clinical trials, regulatory filing, life cycle management, and strategic initiatives in business and development and clinical drug development. Dr. Setnik is also an adjunct professor at the University of Toronto, Department of Pharmacology and Toxicology, and the Managing Director of the Cross-Company Abuse Liability Council, and Chair of the Clinical Pharmacology Community of the Drug Information Association. She is an active member and speaker at various congresses, including the College on Problems of Drug Dependence, and is actively engaged in many aspects of abuse potential assessment, including development of patient-reported outcome instruments and contributing to the post-marketing surveillance studies. Dr. Setnik has a PhD in pharmacology and the collaborative program in neuroscience. We invite the audience to submit any questions you may have for our presenter throughout the webinar using the Q&A widget to the right-hand side of your screen. We will pose your questions to Beatrice in a live Q&A session after the presentation and then follow up any outstanding questions offline. Thank you again for joining us today, everyone. I'll now hand over to Beatrice to start today's presentation.
Hello, and thank you for joining. Today we're going to be covering some of the important considerations for early phase CNS drug and cannabinoid development. Just a brief overview of Altasciences. We're an advancing drug development company from lead candidate selection to proof of concept with over 25 years of expertise in drug development. Our company has specific CNS drug research and abuse dependency evaluation expertise. With locations in the U.S. and Canada, we have state-of-the-art facilities including controlled smoking and vaping chambers. We are the only CRO with permanent driving simulators within North America. Our facilities are located in Seattle for our preclinical work, and our clinical work takes place in Kansas City and Montreal, Canada, with bioanalysis and research services in Laval, Canada. For today's session, you will learn about the regulatory requirements for CNS drug development and cannabinoid development. You'll understand what preclinical studies may be needed to support your application, understand which clinical studies may be needed to support your regulatory submission, and discover the specialty studies that are used strategically to evaluate your risk-benefit profile. CNS active drugs and cannabinoids have unique attributes and need to have special consideration for things such as cognitive impairing and enhancing effects, potential reinforcing effects which may lead to abuse, potential ability to cause physical dependency and tolerance, and some may cause additive effects, both pharmacokinetic and pharmacodynamic, when combined with drugs or alcohol. There are important regulatory considerations to take into account when developing CNS products. Several FDA guidances can be of assistance to you during your course. One is the assessment of abuse for potential for drugs, Another is the evaluation of drug effects on the ability to operate a motor vehicle, and there is also a specific guidance for those developing botanicals. We will be covering most of the information here today. First, we will begin with driving simulation studies. According to the FDA guidance, the driving impairment issue is a public health priority, and there's a systematic effort to identify drugs that increase the risk of motor vehicle accidents, abbreviated MVA, and reduce those risks. It is understood that patient self-perception is not adequate for evaluating the presence or degree of driving impairment, and oftentimes in clinical trials it's important to include objective information to determine how a drug affects driving ability. Many people ask what drugs need to be evaluated for driving impairment. Such drugs that are intended for chronic, including chronic intermittent use by adults who drive. These may include both psychoactive and non-psychoactive drugs. For example, you may have impaired consciousness from a hypoglycemic reaction to a glucose-lowering drug, or impaired vision from a mydriatic drug. There are drugs that have the potential to improve driving performance by decreasing somnolence, and some might increase risk-taking, for example, stimulants that may impose aggressive driving. If there are drugs that are intended to be taken at night, they may have residual daytime effects. Driving studies may also be needed for drugs that are already approved for marketed use, particularly if you may be proposing a different indication, a different dose, a different dosing schedule, or perhaps a new patient population where there's insufficient information about how the drug may affect driving. Important safety information related to driving impairment is often presented in drug labeling. For example, here is an example of Ambien. In section 5.3, you see here in bold that there are warnings about sleep driving when taking the drug. In section 5.5, the label does outline caution against using hazardous equipment or driving a vehicle under the influence of Ambien. The FDA generally endorses a tiered approach when assessing driving impairment. That it does include standardized behavioral assessments that are implemented early in drug development. This would include assessments of pharmacology, toxicology, epidemiology, and clinical standardized behavioral assessments. The tests used in drug development should have a high sensitivity for impairment. While you move later into your development phases, those studies should clarify what the clinical relevance of those earlier findings may be. Some of the important functional domains of testing include areas such as alertness, arousal, wakefulness, attention and processing speed, reaction time and psychomotor functions, sensory perceptual functioning, and executive functions. And as you can see, these domains are relevant to the complex tasks required to drive or operate complex machinery. Early testing in development should emphasize sensitivity over specificity. Beginning even with your first-in-human trials, all drugs, including non-CNS, should be evaluated for CNS adverse events. And those may be important events such as somnolence, agitation, or dizziness, things that may pose issues for impairment. The advantages of testing early in development are that early trials often include higher doses, which allow you for full pharmacological characterization. Subject evaluations allow for correlations between subjective and objective impairment, so it is good to include both subjective and objective measures when you're assessing early in development. It also enables the collection of important safety information that can be used for administering your drug safely in later phase patient trials. As you move on further in development into phase 2 and 3, drugs with potential impairing effects should include pharmacokinetic assessments. Any factors that may affect exposure should be documented, things such as food, time of dosing, or concomitant medications, among others. In addition, it is important to monitor at specified intervals temporal impairment risk. This may be done by open-ended and targeted adverse event collection, reports of motor vehicle accidents or traffic violations during the course of a clinical trial. You will also want to monitor effects that might not have been previously identified, such as impaired executive function or memory. For the driving studies themselves, once you have accumulated data and characterized your CNS drug profile early in development, and accumulating data suggests that there is a potential for driving impairment, then a dedicated driving study may be needed. These types of studies have higher face validity than a more general test of CNS function, and they're typically conducted with either actual motor vehicles or driving simulators, as you see here on the picture on the right. Different driving tests, for example, different scenarios, need to be used to assess somnolence than to assess executive function, such as impulsivity or risk-taking, depending on the type of drug you're using. The CRCDS MiniSim is one of the operating driving simulators that we have at Altasciences. It's a fully standardized, validated PC driving simulator that's optimized for pharmaceutical research. As you can see from the picture here, it has a driving cockpit utilizing a full-size steering wheel and realistic pedals. The actual automotive seating meets the NHTSA standards. It has a tactile transducer that provides driving vibrations to enable a realistic experience while driving. It also contains a multi-speaker audio system and 3-screen wide display presenting a highly immersive and realistic driving environment. The system incorporates MiniSim simulator software developed for the National Advanced Driving Simulator at the University of Iowa's Research Park. It employs the same advanced technology found in the NADS-1 full motion simulator developed for the National Highway Traffic Safety Administration. Some of the methodological considerations for dedicated driving studies. Most will enroll patients who are from the population likely to use the drug to inform about potential disease-drug interactions. However, there may be cases where it's possible to conclude that there are very few differences between the healthy volunteers and patients, and therefore healthy volunteers can be used for these types of studies. It is important to evaluate the effects of the initial dosing and the effects after chronic exposure and test in both environments. Testing should take place at maximum levels of parent and or active metabolite. Determining the time course and extent of any tolerance that develops is also important. Testing at the highest dosed exposures expected to be encountered in clinical use. Because of the early dose ranging in first-in-human studies, it may also be suggested to include some measures of driving simulation early on, even in the first-in-human studies, to give some surrogate indications of whether or not they may be needed and assessed in further on in clinical development. For such studies, a positive control should be used to demonstrate assay sensitivity and to understand the magnitude and duration of impairment. Things like time of day and duration of effects should be documented. The typical design for these driving studies are crossover designed. The design is dependent on questions to be answered. For example, nighttime dosing if there are anticipated next day residual effects, or daytime dosing depending on the onset and duration of effects. As you can see from this table, there is a wide range of sensitivity to sedation across a wide range of medications. This table presents the summary of standard deviation of lateral position, so it's deviations from the road, as a change from placebo. These are the data from 6 independent studies. And as you move down towards the table, it indicates more impairing effects. So drugs like flibanserin have very little effect, whereas the ethanol and benzodiazepines have more of an impairing effect as expected. The endpoint analysis for such studies examined the mean effect on differences from placebos. They conduct a responder analysis that assesses the proportion of patients on drug versus placebo that exceed a predetermined threshold for a clinically meaningful impairment, for example, a symmetry analysis. They also establish the relationship between drug concentration and driving endpoints. Hence, it's oftentimes very important to examine pharmacokinetic and pharmacodynamic effects of your drugs. Now we will move on to the next chapter of evaluations for CNS drugs, and this is the Human Abuse Potential Study. To understand why abuse potential is relevant, it's important to consider the Controlled Substances Act, which was enacted in 1970 in the United States. For this, drugs and other substances that are considered controlled substances are classified into 5 respective schedules. A controlled substance is scheduled based on whether it has currently accepted medical use in treatment in the U.S. and its relative abuse potential and likelihood of causing dependence. Of note, physical dependence is a neuroadaptive process that can occur in the absence of abuse potential. For example, SSRIs can cause physical dependence or withdrawal symptoms when a drug is abruptly discontinued, but are not prone to abuse. The ability of a drug to produce physical or psychological dependence plays a role in the scheduling placement of an abusable drug under the Controlled Substances Act. Here is a table outlining the scheduled drugs in the United States. You'll notice on your left-hand side, Schedules 1 to 5 are presented. As you move from Schedule 1 to Schedule 5, you're moving from the highest abuse potential to the lowest. The main difference between Schedule 1 and 2 is the fact that Schedule 1 substances have no accepted medical use, whereas Schedule 2 do. You'll notice in the 4th column under Prescribing Restrictions that the restrictions lighten up as you move from Schedule 1 to 5. For example, Schedule 1 is only allowed for research. Schedule 2 does not allow telephone prescriptions and no refills are allowed. If you move progressively towards Schedule 5, you can have over-the-counter possibilities. The scheduled drugs here, illustrated in green and red, are examples of drugs that have moved to a lesser schedule or to a more conservative schedule. For example, marijuana, which is not federally legalized, has approved THC compounds. For example, Sesamet, Syndrose, and Marinol are in respective Schedules 2 and 3 because they are approved for medical use in the United States. For example, some drugs have also moved to a more conservative schedule. Hydrocodone, codeine compounds compounded with NSAIDs moved from Schedule 3 to a more conservative Schedule 2, and tramadol, which was previously unscheduled, has moved to Schedule 4. More often, drugs will be rescheduled to a more conservative scheduling, and it is very rare that drugs are scheduled to a less conservative schedule. One example of a differential scheduling is Syndrose, which contains dronabinol. This was FDA approved in 2016 as a Schedule 2. You'll notice that Marinol, that also contains dronabinol, is a Schedule 3. Why the difference? Well, Syndrose is formulated as an oral solution, and it has a conservative scheduling due to the formulation because it is an oral solution versus a capsule as Marinol. It was deemed that this type of formulation may be easily manipulated for abuse by inhalation and other routes. It can be easily evaporated or concentrated into a residue. The formulation also contains alcohol, and it has a large content of product in each bottle. Even though the human abuse potential studies showed equivalent abuse potential at comparable doses, the formulation itself may lead to increased abuse, and therefore a more conservative schedule was applied. Abuse potential. Here is an example of how the FDA and the European Medicines Agency differ in their expectation of how CNS drugs are evaluated for abuse potential. For example, in Europe, in vitro binding and preclinical studies are required. However, in the United States, these studies, plus the human abuse potential study, a physical dependence evaluation in the clinic, and clinical evaluations of cognition and performance are also required. It is very important to make note of these key differences when developing CNS drugs for the American market. When is abuse potential assessed? It is generally conducted for new drugs, including new molecular entities, when the drug and/or its metabolite affect the CNS system. If they're chemically or pharmacologically similar to other drugs with known abuse potential, if they produce psychoactive effects, for example, sedation, euphoria, mood changes, or if they're formulated with abuse deterrent properties. In some cases, as was the case with driving simulator studies, these types of studies may be necessary for a marketed product if it presents an unexpected adverse event profile that includes events related to abuse potential, or if it's being reevaluated for a new route of administration that could affect its abuse potential. So, for example, going from an oral formulation to an intravenous could dramatically change the abuse potential profile of a drug. The human abuse potential assessment is assessed throughout the entire development program. The determination of whether or not you will need a dedicated human abuse potential study, however, is usually conducted after the end of a Phase 2 meeting for a new chemical entity. It is at that point where you will understand more about the pharmacological drug profile, identify the therapeutic dose levels, and identify the maximum tolerated dose. It is during this discussion where you will be determining if dedicated studies for abuse potential and potential driving simulator studies may be needed for your programs. Human abuse potential studies evaluate the likability of a drug using a face-valid population. These are typically single-center, single-dose, double-blind crossover studies. The subject population include non-dependent recreational drug users. These studies, because you are assessing subjective drug effects, include both a placebo and an active control. The controls are always drugs with known abuse potential and include drugs from classes of opioids, stimulants, depressants, sedative hypnotics, cannabinoids, and/or hallucinogens. It's also possible to strategically include negative controls, which are drugs that may be similar to your compound but are not scheduled and don't have abuse potential. The pharmacological challenge is a phase of the study that's included to ensure that subjects are not physically dependent on the study drugs. That means that they are not going to go into withdrawal if they are suddenly abruptly discontinued from the recreational drugs that they use, and that they are sensitive to the active control. In particular, with those folks developing cannabinoids, oftentimes the studies will include positive controls which may be inhaled, smoked, or vaped. These types of studies required controlled smoking chambers, and the consideration of staff exposure to exhaled or releasing smoke or vapor must be carefully considered. In the pictures here are smoking rooms that are at our facilities. As you can see, they are well ventilated and have a two-way mirror and communication system so that staff can communicate with the subjects during the drug administration. Human abuse potential studies include both subjective and objective measures to evaluate the drug effects on cognition, and motor skills. These are typically done on computerized testing administered on tablets for ease of use. What you see here in this picture is the primary endpoint for a human abuse potential study, which is the drug liking. In this scale, the question is asked, at this moment, my liking for this drug is, and it is presented on a 0 to 100 point visual analog scale, where you have strong disliking on one end and strong liking on the other. Subjects will simply move the cursor along how they feel at that moment in response to the drug. Here is an example of abuse potential data for Epidiolex, a recently approved cannabidiol. This study compared cannabidiol in relation to dronabinol, alprazolam, and placebo. What you see here is drug liking presented on the 0 to 100 visual analog scale over time. The placebo, represented in orange, and the CBD, represented in different shades of blue. As you can see, they have very low effects compared to the positive controls alprazolam in gray and dronabinol in fuchsia and red. The data from human abuse potential studies are represented in the drug label. For example, Section 9 covers drug abuse and dependence. In Section 9.2, in this case, this is a sample label of Epidiolex, which was recently approved and received Schedule 5 status. As you'll recall, Schedule 5 is the lowest on the 5 schedules, representing the lowest abuse potential. Section 9.2, and what you see here in bold, are the results of the human abuse potential study that I presented in the previous slide. Abuse and dependence potential evaluation require the evaluation of data collectively reviewed by several agencies, and those will determine drug scheduling. The FDA, the NIDA, HHS, and the DEA all play important roles in reviewing data and determining ultimately what schedule a drug may or may not have. The data that is reviewed covers everything across the entire development program, and it's important to understand that. The human abuse potential study is a pivotal part of that, but is one of many pieces that contribute to the data set. Data collected that's considered includes everything from chemistry studies, receptor ligand binding studies, PK studies in both animals and humans, dedicated abuse studies in animals, which include general behavioral observations from any of the safety pharmacology studies, and the dedicated drug discrimination and self-administration study, which is required for abuse, and the physical dependency studies conducted in animals. Then we move on to important data from abuse-related studies in humans, which include both the human abuse potential study that we just covered and the physical dependency study that I will discuss shortly. The adverse events collected from the entire clinical program are also evaluated for key signals of abuse and physical dependence. Information related to overdose, both intentional and accidental, during clinical studies are also looked at, as well as the assessment of the incidence of abuse during clinical studies. So it's important to be mindful that all of this information will be reviewed as part of the scheduling determination, and there are ways to organize this information in an effective way in your NDA. This can be achieved by an 8-factor analysis, which is what the FDA uses to determine drug scheduling for your compound. Now, sponsors are required to set out a proposal for scheduling. Doing the 8-factor analysis yourself proactively as part of the NDA submission allows you to organize all of the data I presented in the previous slide in a comprehensive fashion that supports your proposal for whether or not the drug should be scheduled, and if so, what schedule it should fall on. Our experts here at Altasciences can help you strategically compile the 8-factor analysis and guide you through these important regulatory decisions. The 8-factor analysis essentially examines the history of the drug, the relative and actual potential for abuse, the safety risks, and the risk-benefit profile. Physical dependency is a state of adaptation that's manifested by a drug class. Specific withdrawal syndromes that can be produced by abrupt cessation, rapid dose reduction, reducing blood levels of the drug, and or administration of an antagonist. The physical dependency assessment has been rigorously defined in the FDA Guidance on Abuse Potential. It requires both preclinical and clinical evaluation. It helps to inform drug scheduling decision, and it also provides relevant safety information in Section 9.3 of the product label. Various symptoms of withdrawal can be found when a drug is abruptly discontinued. These can range from mild to serious. New withdrawal symptoms, which are common to many CNS drugs, include symptoms such as nausea, anxiety, headaches, decreased concentration, irritability, agitation, tremor, sleep disturbances, etc. Some new withdrawal symptoms may be specific to certain CNS drug classes. For example, lacrimation, rhinorrhea, and sneezing are typically seen with opiate withdrawal, whereas electric shock sensations, confusion, and myoclonus are typically seen after abruptly discontinuing SSRIs. Usually, these types of withdrawal symptoms are transient and reversible, but there are major complications of abrupt withdrawal, which can occur, such as seizures, suicidality, and psychosis. Onset of withdrawal symptoms depends on the duration of action. Needless to say, the agencies are very particular about this type of data because they contain very important safety information that's very important for the safe conduct of your later phase clinical trials, as well as important patient information once the drug becomes marketed. The physical dependency data helps inform physicians, patients, and drug abusers about the consequences of abrupt withdrawal, the potential need for tapering or switching to an alternate drug with a similar mechanism of action following discontinuation, any of the specific serious adverse events that could be caused if a drug is abruptly discontinued, or the potential risk of dependency, sustained use to avoid withdrawal symptoms. That would be the case, for example, in opiate users who abuse the drug and then have a difficult time staying off the drug because of the physical dependency and the dope sickness they feel. Abrupt drug discontinuation can lead to various clinical manifestations. For example, there are acute withdrawal symptoms, which include new signs and symptoms which do not represent the relapse of the underlying condition. There are rebound effects, which is a recurrence of symptoms of the treated disorder in patients, but that's usually more severe than it was before the treatment. There are also post-acute withdrawal disorders, and those are things that can be appearance of symptoms that are related to emerging new disorders, and these can be things like tardive dyskinesia or protracted insomnia. There are many classes of drugs that are known to cause withdrawals. For example, scheduled drugs such as opioid, benzodiazepines, stimulants, cannabis, testosterone, ketamine, and a variety of also unscheduled drugs that can cause withdrawal. For example, antidepressants, anti-Parkinsonian drugs, antipsychotics, beta blockers, and corticosteroids. The assessment of physical dependency requires both a preclinical assessment in animals and a clinical. The clinical portion of the dependency evaluation is usually conducted in phase 2-3 trials after the study drug is terminated. This is often overlooked by sponsors and is an important element of designing late-phase trials. It requires pre-planned assessments and visits during the discontinuation period, which are often overlooked if they are not proactively placed in the protocol. We've often encountered many sponsors who have not implemented such evaluations, which requires then further discussion with the agencies. The designs are typically done at the end of a phase 2, phase 3 trial. However, there may be exceptions if, for example, a healthy volunteer dedicated study may be needed if patients cannot be safely discontinued off a study drug in phase 2 or 3. This could be because you are dealing with patients that may invoke seizures or psychosis in the case of epilepsy and schizophrenia, for example. When a dedicated healthy volunteer study is needed, it requires a minimum 30-day exposure followed by an observed withdrawal period that's dependent on the half-life. These studies are typically placebo-controlled and double-blinded. And following study discontinuation, there's an assessment of adverse events, and validated drug withdrawal questionnaires are implemented. We also assess rebound effects in cases where a patient population is used. But it is important to plan for these types of studies because they are becoming very important in the evaluation of drug and abuse dependence. There are several validated withdrawal scales that can be implemented either in a dedicated physical dependency study or at the end of a phase 2, phase 3 trial. And the withdrawal scales are dependent on the class of drugs. As I mentioned before, different classes of drugs can have different profiles of withdrawal symptoms. There are dedicated opiate, benzodiazepine, stimulant, cocaine, marijuana, SSRI, and a general discontinuation emergent and symptoms checklist. The drug labeling also has important information on dependence. If you look at section 9.3, here is an example of Epidiolex. In this case, this drug did not show physical dependency and therefore has wording in the labeling to reflect that absence of symptoms. On the other hand, some drug labels with drugs that do produce physical dependency will often have informative labeling in section 9.3. For example, Oxecta, in the bold here, talks about the types of withdrawal symptoms that are observed following study drug discontinuation. This enables physicians and patients to ensure an appropriate withdrawal following the cessation of therapy. Now we will move on to some additional considerations for CNS drug development. There are other aspects of cognitive function that may also be important to evaluate during your CNS drug development process, and that may be things that are related to attention, memory, visual perceptual functions, sequencing functions, logical problem solving, psychomotor speed and coordination, simultaneous information processing abilities, and executive functions. These may be in and above effects that may be beyond impairing effects of CNS drugs. These may be actual effects that are related to the efficacy of the drug itself. Another important element when evaluating CNS drugs is the risk for suicidality and ideation. Some drugs, for example, Abilify or other SSRIs, have black box warnings related to the risk of suicidality, of suicidal thoughts and behaviors. There is a specific guidance from the FDA around this issue and how to incorporate these assessments in clinical trials. In conclusion, CNS drug development requires unique risk-benefit evaluations that are unique to CNS drugs and are related to the cognitive function and impairment, for example, driving, abuse and dependence potential, and risks for other events such as suicidality. It is imperative to include these types of assessments early in drug development. This will be very important for your end of phase 2 discussions with the FDA to strategically determine what other studies may be needed to further characterize the safety profile of your drug. Understanding the preclinical and clinical requirements, as we've gone over today, can avoid NDA approvals for your drug. Strategically assessing data in an 8-factor analysis can help support scheduling recommendations. As I've mentioned earlier, it's very difficult and challenging to have a drug scheduled in a certain schedule and then have less conservative scheduling applied later. So it's very important to tackle this early and throughout your drug development process. It is important to understand that we're here to help you. Should you have any questions along the way, we can gladly provide consultation on how to manage your clinical trials, how to ensure that you're assessing the appropriate measures throughout your drug development process, and we have the expertise and in-house capabilities to conduct the studies that we've discussed during today's call. Now we will move over to the Q&A session of the program.
Thank you for that very insightful presentation, Beatrice. I hope our audience found it of great interest and value. If you do have any questions for Beatrice, then please continue to submit them using the Q&A tool on the right-hand side of your screen. We have received a number of questions so far, so we will address some of these now live on air. So our first question is, for human abuse potential studies, how do you select appropriate controls and scales for a new chemical entity if it's not related to known drugs of abuse?
Yes, we encounter this situation quite often with new chemical entities. And what we will do, and this is why it's important to conduct these trials usually a little later in drug development after the end of phase 2 meeting. And that is because we need to understand what the pharmacology looks like. We select the controls, and oftentimes there can be more than one control based on the adverse event profile, for example, and the pharmacology. So, for example, if the drug is causing somnolence and is perhaps related to treating sleep disorders, or the obvious type of positive control could be, for example, a benzodiazepine in those cases. Sometimes drugs may have some euphoric effect and may have a stimulating effect, so an opioid and a stimulant may be required. So it really depends on how the drug works, what the pharmacological effects are, what the pharmacodynamic data looks like, and what the adverse event profile looks like. And the scales are similarly adapted. Drug liking is always the primary endpoint in these types of studies, but additional scales may be selected depending on the effects of the drug. So it's important in these cases to understand, to have enough information collected about the pharmacology as well as the dosing and identifying that maximum tolerated dose. And that's usually that data is collectively evaluated to provide the best suitable design for a human abuse potential study.
Okay, that's great. Thank you, Beatrice. Our second question is, how frequently should withdrawal assessments be made during the drug discontinuation phase of a patient study?
Now, this is a very important thing to consider because it will really depend on the half-life of the drug, when the drug levels are expected to drop, and usually a period of 2 to 3 weeks is needed, and it may be longer if you have a longer half-life. And the frequency really will be dependent on the duration of the drug effect, both the pharmacodynamic effects that are observed, as well as the pharmacokinetic profile. Usually you will want to have some assessments done fairly close to when the drug is discontinued to get the sudden decreases in drug levels as they're dropping and as they come to a decreased amount, a significantly decreased amount. And then you'll want to monitor those effects for some time to see the resolution of those adverse effects as they continue, if they do continue as a result of drug cessation. So it can be dependent. It really does need to be dependent on that, but it is not simply enough to do one assessment. Usually you may need to do more than one to fully characterize the withdrawal symptoms.
Okay, that's great. And question number 3 is, what are the regulatory differences when conducting studies in Canada versus the US? Usually
the Canadian environment is a very friendly environment for clinical trials, and we do a lot of our clinical work up in Montreal as well as in Kansas. In Canada, the sponsors are required, and we do this for them, a clinical trial application, which is submitted to Health Canada and has a 30-day review period by Health Canada. We provide all of the expertise and the provision of those types of packages, but essentially it includes the protocol, the ICF, and the CMC section around the drug, which is usually slightly less than what's in the IND. So if a sponsor has filed an IND, they will have the information needed on hand to produce the CTA package. It's a 38-day turnaround. It is done in parallel with the IRB approval process, and it's quite an expedited process to get studies up and running in Canada. Other than that, if you're shipping a controlled substances, there are import-exports that we also take care of and we receive. All of our facilities are licensed for receipt and shipment of controlled substances in Canada and the U.S., and those activities are also done in parallel to the application processes for the CTA and IRB to help expedite study startup times. So the quality of the study is the same whether you run it in Canada or the US. In fact, there are some advantages in Canada, including tax credits and perhaps sometimes a better value for the currency exchange. So there are some very good reasons why folks do come up to Canada and to conduct studies, including our healthcare system, which is universal for patients as well. And we'd be happy if any sponsors have further questions about conducting trials in Canada. We'd be happy to answer any questions. We do provide the full support services to help sponsors with all of the needs to set up studies up in Canada or the United States, wherever they may decide to conduct the clinical trials.
Thank you very much, Beatrice, for answering our audience questions today. Before we wrap up, do you have any last comments you would like to add?
Yes, I would just really like to emphasize to the audience that proactive planning of your clinical program and your drug development program is so essential, and understanding these guidances and the implications that they have on early and late phase trials is critical. And it takes a proactive approach from the very beginning of drug development to strategically present your data and have more meaningful conversations with the FDA to determine what extent of abuse potential testing you may need to do, of whether or not you may need driving simulator studies. And the more data that you can collect proactively right from the beginning of your program, the better position you will be to both understand the risk-benefit profile of your drug and to understand what studies may be needed throughout your package to avoid very costly delays in your regulatory submission packages. So I would encourage all sponsors to really examine these carefully, and you're not alone. We are here to help. I'd be happy to consult with any clients that are in need of advice on how to best position their programs and what kind of regulatory strategies they may take for their specific drug programs. So I encourage anyone, if they have any further questions, to please feel free to contact me. I'd be happy to discuss any further questions that you may have regarding your specific drug programs.
That's great. Thank you very much, Beatrice. So I would like to once again thank our wonderful presenter, Beatrice, as well as you, our audience listeners, for your time and questions. If you would like to let us know your thoughts on today's webinar, then please feel free to complete our feedback survey in the resource widget or tweet us at neuro_central. Don't forget to visit us at www.neuro-central.com for more webinars. Thank you again for attending, and goodbye.